Okay, hello everyone. Here we are today with a new topic. We're still focusing on the metabolism of lipids. I'm going to remind you we discussed about the digestion of dietary lipids. We discussed about the beta oxidation or degradation of fatty acids to carbon dioxide and water. And now, like we did with the carbohydrate metabolism, we're gonna explain the opposite pathway, like we did for glycolysis versus gluconeogenesis. We're gonna do the same with fatty acids. So after beta oxidation of fatty acids, we're gonna focus on anabolism of fatty acids, and we're gonna talk about the biosynthesis of fatty acids. So in order to start, let's make some kind of a review of the process and some kind of a review of what we are gonna discuss about today. So the major portion of fatty acid in our organism is obtained obviously from diet. If you remember when we discussed about the digestion of dietary lipids, we saw that the dietary lipids are digested, absorbed to enterocytes, resynthesized to 3-acylglycerols as major sources of fatty acids in order to form chylomicrons. Chylomicrons are then transported to bloodstream. The metabolism of chylomicron is going on in the circulation, and hydrolysis of 3-acylglycerols from chylomicron occur, providing significant amounts of fatty acids. Then we saw that fatty acids are entering cells and entering mitochondrion, and they are subjected to beta oxidation to form acetyl coenzyme A. Acetyl coenzyme A can be now used for the biosynthesis of fatty acids, of course, in cases when a cell needs the biosynthesis of fatty acids. But what is very interesting is that the carbohydrate or sugar-rich diet actually yields the formation of significant amounts of acetyl coenzyme A. So remember, carbohydrate-rich diet starts with the digestion of carbs, and a huge amounts of glucose are transferred to blood, from blood into cells. Glucose undergoes glycolysis to form pyruvate. Pyruvate enters mitochondria, undergoes oxidative decarboxylation to form acetyl coenzyme A. So acetyl coenzyme A, of course, it's going to enter the Krebs cycle in order to provide a certain amount of energy for the cell. But in case when a cell does not need an energy, the fast dose certain significant amounts of fast energy from carbs, acetyl coenzyme A can be converted to fatty acids, and from fatty acids, of course, incorporated into 3-acylglycerols, and 3-acylglycerols can be accumulated or deposited in adipose tissues, getting some kind of pool or reservoir of fatty acids for the periods of starvation or for the periods of increased energy needs. So we can conclude from this that actually the major source of carbons for the biosynthesis of fatty acids are actually dietary carbohydrates. Speaking of protein-rich diet, yeah, of course, if you have an excess amounts of proteins in a diet, more than cells need for the protein synthesis, certain slightly increased catabolism of amino acid can occur. This is what we are going to discuss in future. So ketogenic amino acids can be synthesized, and they can undergo further degradation and catabolism to either acetyl coenzyme A or some ketone bodies or some intermediates of the Krebs cycle. So again, driving the whole and directing the whole pathway towards the biosynthesis of fatty acids and subsequent biosynthesis of 3-acylglycerols. But this is a process which is not that pronounced and intensive as the conversion of carbohydrates to fatty acids. because our organism tends to save amino acids, tend to save proteins, and this is going to be discussed later on. So fatty acid biosynthesis occurs predominantly in liver. Location in cells is very well known. We've already mentioned it with beta oxidation. So it's located in cytosol. So all enzymes are located in cytosol. But fatty acids can be also synthesized in some other tissues, but to a lesser extent. So less in mammary glands, adipose tissue, and kidney. We've already mentioned last time that those reactions are mainly reversible to those of beta oxidation, but this time we have the addition of 2 carbon units to an increasing fatty acid chain. To answer the question, which are the major precursors for the biosynthesis of fatty acid, I can say that those are 2 precursors. First, acetyl coenzyme A. But acetyl coenzyme A in its original form is used for incorporation of only first 2 carbon atoms into the fatty acid chain. All other carbon atoms are incorporated from malonyl coenzyme A. So malonyl coenzyme A is considered to be a precursor for the fatty acid synthesis. Apart from those precursors, we also need an energy for the synthesis of new chemical bonds. So like all other anabolic processes, we need ATP for the completion of those pathways. And as like most of the biosynthetic processes which are characterized to be reductive biosynthetic processes, we require some donors of reducing equivalents, and those are going to be NADPH molecules, which we discussed in the chapter of hexose monophosphate pathway. Also, apart from these components, there is one component which is very specific to the biosynthesis of fatty acid, and that is a specific acyl residue carrier, which is ACP protein or acyl carrier protein. So acyl group is going to be attached and transferred to ACP and not to coenzyme A like we had in beta oxidation of fatty acids. So, as I've mentioned at the very beginning, the major portion of fatty acids and carbon atoms for fatty acid synthesis is obtained from sugars, obtained from glucose. So let's discuss a little bit about the conversion of glucose to cytosolic coenzyme A required for the fatty acid synthesis. So you remember the glycolytic pathway throughout which we are degrading glucose to pyruvate. And we know that pyruvate enters mitochondria. And in mitochondrion, as you can follow from the figure here, it can be subjected to 2 different changes. So it can be subjected to transformation to acetyl coenzyme A by the complex of pyruvate dehydrogenase in the pathway of oxidative decarboxylation of pyruvate. Or it can be carboxylated to oxyl acetate by pyruvate carboxylase enzyme. We know from the previous topics that the increased concentration of acetyl coenzyme A actually inhibits the pyruvate dehydrogenase activity. So the fate of pyruvate is redirected towards pyruvate carboxylase reaction and the formation of oxyl acetate. Increased concentration of oxyl acetate and already increased concentration of acetyl coenzyme A in a cell tends to make a reaction between these 2 molecules to form citrate molecule. Then citrate is transported from mitochondria to cytosol, where citrate can be degraded to acetyl coenzyme A, this time cytosolic acetyl coenzyme A, and oxyl acetate again by the enzyme citrate liase. So this is the way how glucose can be converted to acetyl coenzyme A, cytosolic acetyl coenzyme A, which is going to enter the biosynthesis of fatty acids. But these pathways are also important for obtaining the sufficient amounts of NADPH coenzyme for reductive reactions of this metabolic pathway. So if you remember from our previous topics, the major amount of NADPH coenzyme is obtained from hexose monophosphate pathway upon the two irreversible oxidative reactions by glucose 6-phosphate dehydrogenase enzyme, yielding the formation of two molecules of NADPH. But also at the time, I mentioned that NADPH can be synthesized in some other tissues, in some other metabolic pathways, in order to provide NADPH for reductive biosynthesis, and this is one of these alternative pathways. So NADPH can be obtained during the conversion of citrate to pyruvate in cytosol. So I'm going to get back to the figure and do some additional explanations. So when citrate enters cytosol, it undergoes degradation by citrate lyase, and ATP molecule is used for the purpose to give oxaloacetate and acetyl coenzyme A for fatty acid biosynthesis. Oxaloacetate further undergoes reaction which is very well known to us from gluconeogenesis and Krebs cycle. So under the action of cytosolic malate dehydrogenase, which is NAD-dependent enzyme, oxaloacetate is converted to malate. And then malate does not enter the Krebs cycle, but rather malate is converted to pyruvate by malic enzyme, and the reaction is oxidation and decarboxylation. Malic enzyme is NADP requiring enzyme. So if you can see, we get pyruvate, which can enter again mitochondrion and close the cycle, but we also get NADPH coenzyme for the biosynthesis. Regarding the fact that this reaction is something that is new for us, I have written this reaction in the form of molecular structure. So this is malate undergoing decarboxylation and undergoing reaction of oxidation of hydroxyl group to keto group by the action of malic enzymes. Enzyme and NADPH reduce, so pyruvate is reduced, which enters mitochondria again. So we have some kind of problem. I'm talking all the time about cytosolic acetyl coenzyme A, cytosolic acetyl coenzyme A, because if you remember, the biosynthesis of fatty acids occurs in cytosol. But if you know from our previous topics, acetyl coenzyme A is synthesized in mitochondrion. So we have degradation of glucose to pyruvate. Pyruvate enters mitochondrial matrix, and pyruvate is converted to acetyl coenzyme A. So acetyl coenzyme A is found in mitochondrial matrix. The other way, the other thing is beta oxidation of fatty acids, which occur in mitochondrial matrix. So again, acetyl coenzyme A is synthesized in mitochondrial matrix. So from multiple sources, our acetyl coenzyme A has to be transported to cytosol, but acetyl coenzyme A cannot pass the mitochondrial membrane because of the steric problems and issues with the coenzyme part of the coenzyme A part of the molecule. So we have to employ a shuttle system like we did for the entrance and transport of fatty acids from cytosol to mitochondria, and this time we're going to employ tricarboxylate citrate shuttle system, which is composed of several enzymatic reactions. So here we are. We have an acetyl coenzyme A from multiple sources. So it reacts with oxaloacetate from mitochondrion, and the reaction is catalyzed by citrate synthase to form a citrate molecule. Citrate molecule can be transported through inner and outer membrane. On the inner membrane, we have a specific citrate transporter, and citrate can easily pass the outer membrane. In cytosol, citrate can be degraded by citrate lyase, and we can get again acetyl coenzyme A and oxyl acetate, and for the reaction we need ATP molecule. Oxyl acetate is then converted by malate dehydrogenase to malate, and malate can pass mitochondrial membrane and enter mitochondrial matrix again to be converted by mitochondrial malate dehydrogenase to oxyl acetate and close the cycle. Or there is an alternative pathway that malate can be converted by malic enzyme to pyruvate, and then pyruvate can enter mitochondrion via pyruvate transporter, and then pyruvate can be carboxylated to oxyl acetate, and again we are closing the cycle. So if we cannot transfer directly acetyl coenzyme A through membrane, we can do it via citrate and a couple of these subsequent enzyme catalyzed reaction. So now we have sufficient amounts of cytosolic acetyl coenzyme A for fatty acid synthesis. But we mentioned that apart from acetyl coenzyme A, we need one other even more important precursor, which is malonyl coenzyme A. So let's see how malonyl coenzyme A is synthesized from acetyl coenzyme A. So the reaction is represented here in the figure. So malonyl coenzyme A is an immediate donor of 2 carbon units to the growing fatty acid chain. So it's synthesized by the action of enzyme acetyl coenzyme A carboxylase. So acetyl coenzyme A undergoes carboxylation process. Like all other carboxylases, this acetyl coenzyme A carboxylase also require the presence of biotin as a coenzyme, and not only coenzyme, it's a prosthetic group covalently attached to the enzyme. So if you remember, we did it previously and explained it previously. Actually, the carboxylation process is completed in 2 steps. First, carbon dioxide is attached to biotin to form a specific carboxybiotin enzyme substrate complex, and then with the involvement of ATP and its conversion to ADP, carbon dioxide is transferred from biotin to substrate, in this case to acetyl group, forming malonyl coenzyme A. Carbon dioxide is obtained from dissolved bicarbonate ions, and it's very important to emphasize here that this carbon dioxide is not going to be incorporated in the chain of fatty acids. So it's going to be released in one of the upcoming steps, and I will point it out that again. Acetyl coenzyme A carboxylate is the regulatory enzyme, and not only regulatory enzyme, but the main regulatory enzyme. So numerous factors can affect the activity of this enzyme, and I'm going to talk about it a little bit later at the very end. So now that we have cytosolic coenzyme A, we have malonyl coenzyme A, we have NADPH coenzyme, so we have all the ingredients needed for the biosynthesis of fatty acids, so we can start with the steps of biosynthesis. So the biosynthesis of fatty acid is completed by a complex enzyme which is called fatty acid synthase complex. So it is a huge enzyme complex, and it consists of 2 identical subunits, and each of these subunits actually has 7 catalytic activities. Also, each of these subunits has one very specific protein that I've already mentioned about, that is ACP, acyl carrier protein, and that ACP or acyl carrier protein has a very unique structure, which is similar to coenzyme A structure because it contains phosphopentane structure like we had in coenzyme A, and also the similar to coenzyme A, we have an active sulfhydryl SH group, which is going to be the reactive center of this molecule, to which malonyl coenzyme A is gonna bound. Additionally, on the other side of the one subunit, we have again one more active sulfhydryl SH group, but this is the SH group from the side chain of cysteine. So on one side of the subunit, we have ACP with SH group, and on the other side, we have cysteine with SH group. So when those two dimers associate, they are actually organized in a so-called head-to-tail arrangement. So one subunit has, for example, ACP-SH group on the top and cysteinyl-SH group at the bottom, but the next dimer is going to have a cysteinyl-SH group on the top and ACP-SH group at the bottom. So finally, we can conclude that SH group from ACP and SH group from cysteine are closely aligned, which is going to be very important to make those intermediates close to each other and to enable very efficient and fast reactions of fatty acid synthesis. So let's move on to the first step. First step actually describes the transfer of acetyl group from acetyl coenzyme A to SH group of ACP protein. And this transfer of acetyl group is completed by the enzyme acetyl coenzyme A ACP transacylase. So it catalyzes the transfer of acetyl group from coenzyme A to ACP. But immediately after that, acetyl group is moved from ACP to cysteinyl SH group. So despite the fact that this phase and this step is not represented in the figure, this is what we get at the end. So we have an acetyl group attached to SH group of cysteine and SH group of ACP protein released in free form and ready to accept the upcoming malonyl coenzyme A precursor. So the next step, the second step, is actually binding of malonyl coenzyme A to SH group of ACP protein. This binding is catalyzed by malonyl coenzyme A ACP transacylase, meaning transfer of malonyl group from coenzyme A to ACP. So malonyl and acetyl groups are actually, be careful, are attached to different subunits of fatty acid synthase, if you have in mind that head-to-tail arrangement. So this is the first subunit, this is the second one, but opposite ends. So the result is that malonyl residue and acetyl residues are now positioned to be very close to each other, which enables an easy chemical reaction incorporated and described by the step number 3 of the reaction. So these chemical changes are described as condensation and decarboxylation. So now notice that the carbon dioxide, which was incorporated during the synthesis of malonyl coenzyme A, is now released from the system. So it's not going to be the part of the future fatty acid. So this condensation occurs yielding the production of beta ketoacyl group. So actually at the very end of this third step, we're going to have 4 carbon unit, and at the beginning of the synthesis it's going to be the derivative of acetoacetate. The enzyme catalyzing this third stage of biosynthesis is beta or 3 ketoacyl ACP synthetase. Notice that this is a thioester bond, which is a high energy bond. The fourth step, the fifth and the sixth step are actually a series of 3 reactions which are reversible to beta oxidation. So let's describe them. So I am going to remind you, if we had first dehydrogenation followed by hydratation and the second dehydrogenation and theolytic cleavage, now we are going to have hydrogenation or reduction, dehydratation or removal of water, and again the second reduction. So they're going to be opposite to those of beta oxidation. So the fourth step, here it is. It's the first reduction. So keto group from acetoacetyl part of the molecule is reduced to hydroxyl group. Enzyme catalyzing this reduction is beta or 3, ketoacyl ACP reductase, and the donor of reducing equivalent is going to be NADPH. Then we move on. After reduction, we have dehydratation or removal of the molecule of water, thus forming double bond. The enzyme catalyzing this fifth step of fatty acid synthesis is marked as beta hydroxyacyl dehydratase. And the last step, the sixth step of the fatty acid synthesis, is the second reduction. So this double bond is saturated or hydrogenated to give single bond. Donors of reducing equivalents are again NADPH molecules, and enzyme is N-oyl ACP reductase. It's not shown on this figure, but will be visible in the picture on in the figure on the next slide. This acyl group is now transferred again to SH group of cysteine in order to release the SH group of ACP, which is going to be ready to accept the new molecule of malonyl coenzyme A and to start the new cycle. And actually this is explained here in the figure, so this is a unique summary of the process. So let's do it together. So this is acetyl coenzyme A entering the fatty acid synthesis, and here is an attachment of acetyl group to SH group of ACP protein. Acetyl coenzyme A is also involved in carboxylation to malonyl coenzyme A by the action of acetyl coenzyme A carboxylase. So the next step is the transfer of acetyl group from ACP protein to SH group of cysteine, and SH group of ACP becomes free and ready to accept malonyl coenzyme A, which is attached to ACP SH group. So this is the first step. The second step is condensation and decarboxylation to give 4-carbon fragment. And then we have 3 reactions which are reversible to beta oxidation. So we have reduction of keto group to hydroxyl group, dehydratation to double bond, and again reduction to single bond, and 4-fragment acyl group, which is now transferred to SH group of cysteine in order to release the SH group of ACP, which is again ready to accept new malonyl coenzyme A, and the reactions are repeated 2, 3, 4, 5, and so on until palmitate, the fatty acid containing 16 carbon atoms, is synthesized. So what is the energy yield of fatty acid synthesis? This pathway ensures the synthesis of palmitic acid, and all other fatty acids with longer chain are actually modified, additionally modified after the biosynthesis is completed. So for the biosynthesis of palmitic acid, we need one acetyl coenzyme A for the first 2 carbon atoms, and then 7 malonyl coenzyme A's. For each malonyl coenzyme A incorporated, we need 2 NADPHs, meaning 14 NADPH coenzymes in order to get palmitate. But we have to take into account that we had to synthesize malonyl coenzyme A prior to this biosynthetic process. So for the synthesis of 7 malonyl coenzyme A's, we need 7 carbon dioxides and 7 ATP molecules. So when we add these two reactions, the total reactions will show us that for the biosynthesis of 1 molecule of palmitic acid, 14 NADPH molecules are required and 7 ATP molecules. But this is the biosynthesis of palmitic acid. But for the biosynthesis of fatty acids with more carbon atoms, like 18, 20, and so on carbon atom, palmitic acid has to undergo elongation. So palmitic acid is going to be precursor for longer chain fatty acids. And this elongation is completed by the action of enzymes called elongases. This process of elongation may occur in both mitochondria and endoplasmic reticulum. But regardless on which localization it's gonna take place, activation to palmitoyl coenzyme A is necessary. If we are talking about the elongation in mitochondria, it actually it's completed by the addition of acetyl groups, not malonyl groups, so acetyl group, and these reactions are reversible to beta oxidation. The difference is only in the last reaction, because in the last reaction the donor of hydrogen atoms is NADPH and not NADH like in beta oxidation. Meanwhile, on endoplasmic reticulum, we have the addition of 2 carbon units in a similar manner that we described for the fatty acid synthesis. So malonyl coenzyme A is added to elongating chain. But the difference is that the acyl group is attached to coenzyme A and not to ACP this time. Our body also needs some unsaturated fatty acids. So desaturation of fatty acids involves molecular oxygen, NADH coenzyme, and cytochrome B5. This desaturation is completed by enzyme desaturase, and double bond is most often formed at the position number 9. So if you remember the structure of most common fatty acids, most of them apart from arachidonic, they have the first double bond at the position number 9. In human cells, we can make double bonds at position number 6 and at position number 5, and not on the rest of the position. On positions 12 and 15, we cannot make desaturation. So polyunsaturated, the process is described and illustrated here in the figure. So for desaturation at position number 9, we need molecular oxygen, and the reaction is catalyzed by fatty acyl coenzyme A desaturase, and we have reducing equivalents 2 protons for this reaction. So in this reaction, cytochrome B5 is going to change its ferrous state into ferric state, and then it's going to be reduced again by the action of FADH coenzyme, which is going to be reoxidized, and then FADH reduced is going to be regenerated by the involvement of NADH reduced coenzyme. But speaking about polyunsaturated fatty acids, because we cannot make double bonds at the position 12 and 15, those are considered to be essential, and you know very well that those are linoleic and linolenic, and arachidonic acid is considered to be partially essential because we can synthesize arachidonic acid, but only if we have linoleic acid as a precursor, which is going to be the part of some other topics. Speaking about the regulation of fatty acid synthesis, like we had in beta oxidation of fatty acids, we can talk about short-term regulation and long-term regulation. So getting us back to the fact that the major sources of carbons for fatty acid synthesis are actually carbs or sugars. So after carbohydrate meal, there is an increased insulin-glucagon ratio. So insulin is increased, glucagon activity is decreased. That situation, if you remember, leads to the increased formation of cytosolic acetyl coenzyme A, because insulin is going to promote glycolysis, it's going to promote pyruvate dehydrogenase activity, so the concentration of acetyl coenzyme A increases, thus inhibiting pyruvate dehydrogenase activity in the favor of the formation of oxyl acetate. And then we have the formation of citrate, which exits the mitochondria, and we have an increase in the concentration of cytosolic acetyl coenzyme A. Also, an increased insulin-glucagon ratio is gonna affect the biosynthesis of those important enzymes involved in the fatty acid synthesis. So the biosynthesis of malic enzyme is going to be increased. The biosynthesis of citrate lyase is going to be increased. The biosynthesis of glucose 6-phosphate dehydrogenase is going to be increased because it has to supply the cells with sufficient amounts of reducing equivalents in the form of NADPH. Also, we mentioned that the increased concentration of acetyl coenzyme A in a cell causes an increase and accumulation of citrate in mitochondria. So citrate can exit mitochondria and pass into cytosol, and this is also enabled by allosteric inhibition of isocitrate dehydrogenase by high energy levels. So if you remember Krebs cycle, we said that when there is a sufficient amount of energy and signals in the form of increased ATP and increased NADH, isocitrate dehydrogenase is a major regulatory enzyme, is going to be inhibited. So citrate is going to accumulate, which is going to shift the equilibrium towards the passage of citrate from mitochondria to cytosol. Its degradation to oxyl acetate and acetyl coenzyme A, so that cytosolic acetyl coenzyme A can enter fatty acid synthesis. Not to forget the impact of malonyl coenzyme A. In the previous chapter, when we discussed the beta oxidation fatty acid and its regulation, I have mentioned that malonyl coenzyme A, as one of the intermediates of fatty acid synthesis, is an inhibitor of carnitine acyltransferase 1, which means that the passage and transfer of activated fatty acids from cytosol to mitochondria is disabled by malonyl coenzyme A, which means that in the end beta oxidation of fatty acid is going to be inhibited. This is very important because during the fatty acid synthesis we have the accumulation of malonyl coenzyme A. So oxidation is going to, and malonyl coenzyme A is going to inhibit the oxidative processes. So oxidation of fatty acid is going to be inhibited while synthesis is proceeding, and the opposite. Also, regarding in regard with the short-term regulation, we cannot skip the story on the main regulatory enzyme, which is acetyl coenzyme A carboxylase. So we said that it's a major regulatory enzyme, which means that numerous factors are going to affect the activity of this enzyme. So there are both allosteric and hormonal regulators of this enzyme. So this is also described in the figure here. So acetyl coenzyme A carboxylase carboxylate is activated allosterically with citrate. And we have already mentioned that citrate can be obtained by the degradation of glucose from Krebs cycle. So if Krebs cycle is inhibited by sufficient amount of energy, citrate is going to accumulate, and it's going to be a signal to increase the activity of acetyl coenzyme A, carboxylase, and fatty acid synthesis. On the other side, let's go further on to fatty acid synthesis. So the ultimate final product of biosynthetic pathway is palmitoyl coenzyme A. So the product of the pathway itself can inhibit the major regulatory enzyme by the mechanism of feedback inhibition. Let's move further on. AMP, you know very well that AMP, adenosine monophosphate, accumulated in a cell is a signal of low energy level in a cell. So there is an AMP activated protein kinase which is responsible for phosphorylation of acetyl coenzyme A carboxylase, and phosphorylated enzyme becomes inactive. So energy deficiency in a cell manifested by increased levels of AMP is gonna inhibit or inactivate acetyl coenzyme A carboxylase, thus inhibiting or inactivating fatty acid synthesis and promoting their beta oxidation. Move on to hormonal insulin. Actually acts in the form to promote and activate the activity of phosphatase, which is responsible, and you know the mechanism of action of insulin and glucagon as antagonists. So insulin is gonna activate phosphatase activity and dephosphorylation of acetyl coenzyme A carboxylase. So insulin as a signal of high influx of acetyl coenzyme A in cell and accumulation of acetyl coenzyme A, it's going to activate carboxylase towards fatty acid synthesis. And of course, apart from short-term regulation, there is long-term regulation, and I'm sorry, this is a mistake, long-term regulation, which actually means the impact of dietary habits on enzyme synthesis. So prolonged carbohydrate-rich diet containing low fat actually promotes and increases the synthesis of acetyl coenzyme A carboxylate and fatty acid synthetase. On the other side, fat-rich diet or prolonged fasting periods exhibit opposite effects. The presence of polyunsaturated fatty acids is shown to decrease these enzymes' concentration. So anyway, dear students and colleagues, this was all that I wanted to tell you and explain you about the biosynthesis of fatty acid and its connection to beta oxidation of fatty acid and its connection to carbohydrate metabolism. I hope that the story was interesting. So until the next story about lipid metabolism, stay healthy, stay safe, stay smart, and bye-bye.